My name is Johann Mendel, and I was born in July 22 from the year of 1822 in Austria. Known as the father of genetics. I lived in a not urban city until I was 11 years old, and that's when my interest for being a monk started. But my father was against that. During that time, I had depression. But when I turned 21 years old, I finally get the chance to go to a monastery and became a monk. During my time in the monastery, I was so bored that I decided to do experiments with peas because they have many variations and because their offsprings grow easily and quickly. After doing my experiments, I get to different conclusions and stated different principles. The first principle is called principle of dominance. In this principle, it is stated that there are some alleles dominant and some recessive. The second law is called law of segregation, and it says that when gametes are forming, the alleles of each gene separates from each other. The third law is called law of independent assortment, and it says that genes of different traits can segregate independently from each other during gamete formation. I thought that these principles only apply to certain species, but after some research and investigation, it is proved that these principles apply to every species alike. During my time on this earth, people didn't approve my works. They think they were fake and not right. But look at me now. I am known as the father of genetics. I didn't believe in the evolution of Darwin, but instead I believed in that God created everything. People didn't approve what I think, but I didn't matter, and I maintained in my belief.
One of the best ways to predict the outcome of a genetic cause is by drawing a simple diagram known as a Punnett square. This tool uses mathematical probability to help predict the genotype and phenotype combinations in genetic causes. Constructing a Punnett square is fairly easy. You begin with a square. Then, following the principle of segregation, all possible combinations of alleles in gametes produced by one parent are written along the top edge of the square. The other parent's alleles are then segregated along the left edge. Next, every possible genotype is written into the boxes within the square, just as they might appear in the F2 generation. Next, we'll be showing you step-by-step instructions for constructing Punnett square. The first step will be start with the parents. Write the genotype of the two organisms that will serve as parents in a cross. In this example, we will cross a male and female osprey or fishhawk that are heterozygous for large beaks. They each have genotype capital B and lowercase b. The second step will be figure out the gamete. Determine what alleles will be found in all of the possible gametes that each parent could produce. The next step will be line them up. Draw a table with enough squares for each pair of gametes from each parent. In this case, each parent can make two different types of gametes, capital B and lowercase b. Enter the genotypes of the gametes produced by both parents on the top and left sides of the table. Next, fill in the table by combining the gametes' genotypes. Figure out the results. Determine the genotype and phenotype of each offspring. Calculate the percentage of each. In this example, 3/4 of the chicks will have large beaks, but only one half will be heterozygous for this trait.